Imagine being able to peer inside the human brain or heart in all its glorious detail without making a single cut. That's exactly what a team of Chinese scientists has achieved, and their discovery could shake up the future of medical imaging and disease diagnosis as we know it. But here's where the conversation gets really interesting. Their method, relying on something called ionic glass, can turn organs like the brain or heart see-through, preserving their natural structure and unlocking a sort of 3D X-ray vision. For decades, researchers had only one way to map the intricate networks inside an organ: slice it into wafer-thin sections and examine each one under a microscope. While this does reveal details, it destroys the whole organ architecture, making it incredibly hard to understand how everything works together in 3 dimensions. So why does transparency matter so much? Well, most biological tissues are naturally opaque. They block light and resist the entry of fluorescent dyes, which are often used to make specific cells and molecules visible. Previous techniques for clearing organs would sometimes cause swelling, shrinkage, or loss of vital microscopic detail. Freezing methods, too, risked damaging samples with ice crystals, limiting their usefulness for in-depth research, and this is the part most people miss. The Chinese team took a bold new approach, using ionic liquids, special chemical solvents that stay liquid even at low temperatures. When organs are treated with these, they enter what's called an ionic glassy state. Here's what happens. The tissues become transparent, and microscopic structures are preserved and undistorted. Organs treated this way can be kept cold for extended periods without the risk of ice crystal damage. Notably, fluorescent dyes become far more powerful, shining 2 to 30 times brighter, making it possible to detect even faint biological signals that would otherwise go unnoticed. In their experiments, the scientists successfully mapped how human neurons are connected and spotted subtle differences in impulse control compared to animal brains. By making rare proteins and complex neural pathways visible, they open the door to creating full 3D maps of entire organs, something that researchers in medicine and biology have dreamed of for years, but always found out of reach with standard technology. The potential impact? According to sources like the South China Morning Post, this breakthrough could revolutionize precision medicine and pave the way for smarter diagnostic tools. Tsinghua University, a key player in the research, even dubbed their technology X-ray vision for tissues. They have also developed a complete, streamlined approach for prepping samples, staining them, and visualizing organs in 3D. But is all transparency beneficial? Could revealing every layer of organ structure open up new controversies over privacy in medical research, or bring up ethical dilemmas about human enhancement? And do you see potential issues with the widespread use of such technology? Share your thoughts below. Do you think the promise outweighs the possible pitfalls? Or are there hidden risks we should be talking about?